Hello, my name is William Myers, and I will be talking to you today about the computational work I'm doing remotely with the Fredolino Lab at the University of Michigan. So, we're all probably familiar with the idea of bacteria being pathogens, but they are also used as research and industrial tools. But whatever the context, optimal performance of bacteria is dependent upon their phenotype. And for bacteria, their phenotype is mostly a result of how they regulate transcription of their genome. In addition to the more familiar example of transcription factor proteins, bacteria also utilize nucleoid associated proteins to regulate gene expression and manage chromosomal architecture. Interestingly, when you take a global view of protein bound DNA, you start to see these long regions of the bacterial genome that are packed with nucleoid associated proteins and transcription factors. This curious feature of bacterial genomes, which have been compared to eukaryotic heterochromatin, are referred to as extended protein occupancy domains, or EPODs. More specifically, we define EPODs here as genomic regions a kilobase or longer that have very high and sustained protein occupancy. And typically, EPODs silence the genes that they cover. The broad theme of my work is investigation of protein occupancy landscapes, especially in the context of EPODs, to inform our understanding of how bacteria regulate their genome in response to conditions of interest. For this poster presentation, we'll be looking at protein occupancy in relation to DNA methylation in E. coli. So, in E. coli, DNA methylation is involved in repairing DNA replication errors, chromosomal replication, and transcriptional regulation. The two methyltransferases that do the majority of DNA methylation in E. coli are known as DAM and DCM, which each have their own target motifs. Here we are working with deletion strains of these methyltransferases, and the different methylation states at the target sites for each strain are shown in this figure. Here we can see an example of how protein occupancy blocks methylation of DAM target sites in the promoter region of an operon that is associated with virulence in pathogenic E. coli. Now, this is a local example of how protein occupancy inhibits or allows DNA methylation. But what if this kind of regulatory crosstalk between protein occupancy and DNA methylation happens across the whole genome? That is the question we are trying to answer with this work. So, to figure this out, we can first compare differences in EPOD composition between strains. So, here we are looking at the number of EPODs in the y axis strain that do not overlap with any EPODs in the x axis strain. What we can take away overall from this figure is that since EPOD composition is changing a lot between the strains, this indicates that protein occupancy overall is changing a lot between strains. But then we want to see if these changes in protein occupancy are associated with the methyltransferase target sites. Basically, we looked at the distribution of protein occupancy at the methyltransferase target motifs. Now, if there were changes in occupancy local to these sites, we should see varied distribution between the strains. However, you can see here that the distributions between strains are pretty much very similar. And so overall, this, in addition to other results shown in the poster, strongly indicates that there is not a global trend in regulatory crosstalk between protein occupancy and DNA methylation in E. coli. Our analysis so far concludes that this is not a common regulatory feature. And so the future direction of this work looks to instead characterize the specific pathways that are differentially regulated in these methyltransferase deletion strains. We can do this with approaches such as iPage, which identifies the pathways that genes in the EPODs for each strain belong to. And so rather than looking for a global regulatory pattern, we can now use the same data to identify the specific genes and pathways affected by occupancy changes in our mutant strains. This could identify more cellular processes affected by DNA methylation, and it could even reveal previously unknown examples of the seemingly rare cases where DNA methylation and protein occupancy interact to impact gene expression. So there's a lot more to this story if you would like to check out my poster, and I encourage you to come ask me any questions. Thank you for listening.